Hello everyone, my name is Nicolle Paola Gómez León and I'm going to talk about the article Naringin alleviates pneumonia caused by Klebsiella pneumoniae infection by suppressing NLRP3 inflammation. Well, Klebsiella pneumoniae is a gram-negative bacterium belonging to the Enterobacteriaceae family, which has been considered an opportunistic pathogen, primarily infecting hospitalized and immunocompromised individuals. In the lungs, KPN, Klebsiella pneumoniae, can release virulence factors like lipopolysaccharide and immune cells such as alveolar macrophages and dendritic cells, triggering them to secrete inflammatory factors to resist bacterial invasion. However, excessive of these inflammation factors due to immune imbalance can lead to pneumonia, acute lung injury, and other respiratory diseases, often causing organ damage and sepsis. In this graph highlights the significance of NLRP3 inflammation that we have here and then naringin. NLRP3 inflammation found in cells of the innate immune system can activate inflammatory cytokines, and the naringin, derived from Exocarpium C3 grandis, exhibits anti-inflammatory properties suppressing airway inflammation and neutrophil infiltration. Which brings us to the main objective of this article, that was simulate pneumonia induced by KPN infection in mice to investigate the therapeutic effect of naringin on bacteria-induced lung inflammation. Now I'm going to explain you the materials and methods that they use in the article. First, we have the quantitative real-time PCR, that is a molecular technique used to accurately quantify the amount of genetic material in a sample. By selectively amplifying target DNA or RNA through controlled thermocycles, it generates a fluorescent signal proportional to the initial genetic material, enabling precise concentration measurement. In this case, was employed to identify gene expression levels of interleukin 1b, NLRP3, and caspase-1 in total RNA samples extracted from lung cells or tissues. Then we have the Western blotting that involves identifying the presence of a specific protein in a sample by separating it through electrophoresis and then binding it with an antibody for identification. In this case, the technique was employed during an evaluation of lung KPN to determine its foundation and to identify the proteins present in the sample. And now we have the immunofluorescence staining, that is a technique that allows us to know the distribution or location of a specific protein in cells or tissues using fluorochrome conjugated antibodies. In this case, they use it to know the subcellular distribution of a specific protein, that was protein P65 in the MHS cells after a specific treatment. And finally, we have the co-immunoprecipitation, that is a technique that detects interactions among proteins and between proteins and other cellular components. They use a protein-specific antibody to selectively bind to the protein of interest within a mixture of cellular components. In this case, they use it to know the direction of the ASE protein with MHS cells. We have the results. In the first image, we have different lung tissues in different contexts, but all of them with hematoxylin and eosin. We have the control group, the mother group, but the three last of them are the mother group with different NAR concentrations. Why? Because they wanted to evaluate the therapeutic potential of NAR in mouse lung pathology. So the control group have all the lung and alveolar structures in optimal conditions, but they noticed that the mother group had damage to the alveolar structure, leakage of inflammatory cells into the bronchioles, and dilated and congested blood vessels. So they increased the concentration of naringin in the mother group, and they noticed that the damage of lung tissues decreased. NLRP3 is mainly evaluated in this graphic. So it is observed that its expression decreases in the absence of KPN and NAR. However, the exclusive presence of KPN increases the expression of both NLRP3 and the P65 phosphorylated protein. But when NAR is found together with KPN, a decrease in the presence of NLRP3 is observed as a higher concentration here of the NAR, suggesting that NAR reduces its elevation and expression. Now we have these results. In these two pictures, they evaluated the same thing, the concentration of P65 protein in nucleus and cytoplasm. So in the first picture, we have the blue color and the green color. The green color corresponds to the concentration in cytoplasm, while the blue color corresponds to the nucleus concentration. So, considering the comparison between model and control, it is evident that in presence of KPN, the translocation of P65 is increased in nucleus, but in presence of NAR, the expression of NLRP3 is reduced because NAR inhibits the nuclear translocation of NFKB. In figure B, we have the Western blotting. After the infection of cells with KPN, they observed gradual decrease in the concentration of the P65 in cytoplasm but increased in nucleus. But where it was observed, P65 expression did not remain the same when adding a concentration of NAR, because NAR decreased the concentration of P65 in nucleus. Finally, we have the last result that factors related to NLRP3 signaling pathways in MHS were elevated. So compared to the control group, KPN was observed to increase the expression of different factors like interleukins. However, by adding concentrations of NAR that we have here, increasing the concentration, the expression of these factors can be mitigated, suggesting that NAR is capable of mitigating this activation of the NLRP3 inflammasome. Now we have the discussion, and we have three authors. Franklin said, dimers formed by AUC oligomerization play a linking role in NLRP3 inflammasome complex proteins. They agree. Mangan said, as a result of the critical role of NLRP3 in inflammatory diseases, it is recognized as a key target for drugs to alleviate inflammation. They agree. And finally, Archen said, regarding the treatment of lung diseases, there have been serial reports demonstrating the remarkable pharmacological activity of NAR through different lung diseases models. They agree. And now we have two conclusions. The first one is, molecular biology tests allow for the precise and specific identification of genetic infections and carcinogenic diseases. This facilitates early and accurate diagnosis, which in turn allows appropriate treatment to be initiated in a timely manner, thus improving patient survival rates and quality of life. And the last one is, understanding the molecular basis of disease through molecular biology testing has enabled the development of targeted therapies of precision medicine. This leads to be a more effective and personalized treatment. Thank you.